Good afternoon and welcome, everybody. We'll be waiting 1 or maximum 2 more minutes to give all participants time to join this meeting, and then we'll commence right away. So, good afternoon and a very warm welcome to the next BIOCTANE webinar in our webinar series. I'm glad that I can see so many people of you joining this afternoon for today's topic on how to combine fermentation and microbial electrochemistry principles to optimize the production of biohydrogen and platform molecules. And I'm happy and honored to have Eric Trably here on board for today's topic. He is the research director at INRAE, the Environmental Biotechnology Laboratory in Narbonne, France. And he will show us not only his expertise, but also the different aspects and the context of today's topic. Just a few words on where it is located within the BIOCTANE process. So you might know that we're using organic waste as a feedstock, and then we'll be using dark fermentation, so a mixed microbial consortium to convert the waste feedstock towards platform molecules. So this is more or less the context. In organizational terms, you are invited to ask questions at any time. You can do so either by writing them in the chat or in the Q&A function. And if you feel like it's easier to explain them, you can also raise your hand, and we'll be allowing or offering you the opportunity to speak at the end of the presentation. Then it would be nice if you could briefly mention 1 or 2 sentences about your background, because that simply helps in like setting the answer and making it towards what you probably want to know. So having said that, without further ado, Eric, the stage is yours.
Thank you, Gunnar. Thank you for this very nice presentation, introduction. So good afternoon, everyone. So my name is Eric Trably. So I'm research director at INRAE, and I'm participating to the BIOCTANE project, as Gunnar mentioned. And today I will present how to combine fermentation and microbial electrochemistry principle to optimize the production of biohydrogen and platform molecules, essentially from waste. So as Gunnar presented, so in the BIOCTANE project, change my pointer. Yeah, in the BIOCTANE project, we are converting first organic waste to carboxylic acid here and hydrogen and CO2 with the first module of dark fermentation coupled with bioelectrochemical polishing. So the subject of my presentation today is to give you more information on the dark fermentation, what are the challenges in dark fermentation, and why we couple with a bioelectrochemical polishing system to optimize this conversion to hydrogen and carboxylic acid. And the remaining solid phase will go to another thermochemical conversion process, hydrothermal gasification. And all these products will be further converted by our colleagues from Hamburg to precursors for jet fuels. So let's focus on dark fermentation, so microbial process and bioelectrochemistry. So first, I would like to present some context of the research. And so this project, BIOCTANE, is really interdisciplinary. So we are the biological part of the project. So, and this part belongs to a global research area dealing with treatment and valorization of waste. So is what I'm going to present. And I will make some focus on biohydrogen because this is a typical product that we can get from waste, and also biomolecules. And in a second step, I will give you more information, more details on dark fermentation and why we coupled with microbial electrolysis cells. So what are the principles, what are the challenges today? So first, the context. So for a long time, anaerobic digestion or composting were two biological processes that were used to treat waste and effluents. So mostly, for a long time, anaerobic digestion in particular was used to treat waste. So we have to keep in mind that is a treatment process first. Treatment means reduction of the organic content of the waste and also reduction of the pollution. And so we got treated end products. For the past 20 years approximately, it tends to be anaerobic digestion, a valorization process, meaning that on top of the treatment, we try to produce some valuable compounds, molecules like methane, a bioenergy that can be used and that can be converted to electricity, to heat, can be injected in the grid, in the natural gas grid, because methane is natural gas, and also can be used as a biofuel. And also now we produce a digestate that can be used, that can be spread on agricultural soil or can be composted and then commercialized. So that's a today trend. And so as a research institute, what we want is to develop what we can call the anaerobic digestion 2.0, so version 2, which is also named environmental biorefinery. Under this framework, we try to deal with multiple sources of waste within a territory. So we try to combine all these type of waste: urban organic waste, waste sludge, agricultural residues, agro-industrial residues, and effluents, animal waste, and new biomasses, and try to treat first, but also valorize to molecules that can contribute to the bioeconomy sector. So, of course, bioenergy, methane, new energy like hydrogen that I'm going to present, and also platform molecules. So these biomolecules or platform molecules can be produced by fermentation, and some of them can be used as precursors for bioplastics, or in the BIOCTANE project, precursors for jet fuels. But together with this, we produce fertilizers, which is an important topic, to replace the fossil fuel based fertilizers. So the digestate is rich in carbon, nitrogen, phosphorus, and potassium. And also an increasing topic is the water reuse in irrigation. And altogether, we try to optimize the bioprocesses to not only to optimize the conversion and the valorization of the organic matter, but also the treatment. So all of these molecules has to be under sanitary and environmental constraints, so with a low impact for the environment and human health. So is what we are investigating. And in the BIOCTANE project, we are investigating these two products first from waste, and we used food waste as a model waste because it's rich in soluble sugars, rich in carbohydrates that can be converted to this molecule, and also because it's a new stream of waste that could be collected and valorized in Europe. So why do we focus on these two molecules? So first, I would like to make some to give you some basic information about hydrogen and why biohydrogen. So hydrogen is a new energy vector, so it's highly abundant everywhere. It has a high energetic density, and when it's combusted, it produces low greenhouse gases emission by combustion. So it's an important vector for the energy transition. So you have probably heard a lot about hydrogen, but the main limit is a low volumetric density. So it has to be used under pressure. And the storage and transportation are energy consuming. So we have a strict regulation for production and storage. And mainly hydrogen, that's a key point, is presented in a complex form, and especially in organic matter. So hydrogen production today requires extraction and conversion process. So we know a lot about electrolysis. So what I'm going to present is also we can extract hydrogen and molecules from the organic matter. So hydrogen market is a real market. So it's about 74 million tons per year. It's already produced, mostly from natural gas. In EU is about 10 million tons per year, and in France 1 million tons per year. So it's a real a big market already. But as you can see here, is mostly used as chemical reactant in the refinery, so for gasoline, to produce ammonia. So is a key molecules to produce ammonia as a fertilizer. So 30% of hydrogen is converted to ammonia, but also for solvent in different way. So methanol, a big solvent, but other chemicals. And if you look at transportation, it's only few percent that is dedicated to transportation. And so what is expected is in the future, and if you look at the transportation now, it's becoming a reality. So just few examples here in France is you have some high taxi in Paris. So it's about a float of around 600, 700 vehicles fed with hydrogen. So it's some good cars with low greenhouse gases emission. Also some trains are operated with hydrogen where there is no electricity. And by 2035, Airbus is planning to develop new planes running, operated with hydrogen. So it's becoming a reality. And it's expected to increase by a factor of 10 from 2020 to 2050, the quantity of hydrogen that will be used in the transportation sector. So here you have an example. So I would recommend this report made by the Hydrogen Council in 2017, but where they projected the hydrogen demand by 2050. And if you look at the values here, it's about 10 times the energy demand will increase in hydrogen by 2050. And it's not only transportation here in blue, but it's also for power generation, buffering, and decarbonation of the industry. So in industrial energy building up and power. So we will need green hydrogen. And for that, we think that bioprocesses can contribute to this new decarbonation domain. Indeed, hydrogen is produced mostly by natural gas, oil, and coal reforming. So if you look at the distribution of hydrogen, how it's produced, only 10% of hydrogen is produced by electrolysis now, and mostly is a conversion of fossil fuels at 90%. So as a research institute, we try to develop new area and new green area. So if you look at the colors of hydrogen, just briefly, green hydrogen, when it comes from renewable sources like wind sources, solar, and biomass also. And you have different types of hydrogen. I would like to show you that blue hydrogen, it's not hydrogen coming from electrolysis, it's just natural gas reforming and carbon capture is what we call blue hydrogen. But most of the hydrogen is gray, brown, and black with a high carbon footprint and high greenhouse gases footprint. So highly detrimental to the environment. So from waste, what can we do? So there is a very interesting paper published in 2015 where they compared the life cycle analysis of the different technologies to produce hydrogen. And if you look at these two parameters, so one is a global warming potential in blue and the acidification potential in red. And if you look at also, if we look at the technologies, this one is electrolysis. So there is some, depending on the source of electrons, that can be an impact here on the global warming. The thermochemical processes, the natural gas reforming with the highest impact. And just to show that biological processes have the lowest impact because it's a natural process and it doesn't require a lot of energy. This is true for biohydrogen, but it's also true for biomolecules. The life cycle analysis of these molecules compared to other processes is low. At the same time, the productivities of bioprocesses is lower than chemical processes. So, and we have specific challenges for biological processes. So what I would like to show you now is what are the main principles, but also the challenges of the biological processes for each step, the dark fermentation and the MEC. So a few years ago, in 2016, we made a literature survey, a review of all the literature dealing with anaerobic processes in mixed culture, especially in fermentation. So it was about 2,600 articles, and then we focused only on methane, pure culture. We removed, sorry, the methane with only methane, so anaerobic digestion articles, pure culture also because we wanted to focus on waste. So a mixture of microbial communities, and we removed the review articles. So we focus on 860 technical articles. So here you have the final paper. And what is interesting is if you look at the technologies, mostly we have dark fermentation that can be coupled with anaerobic digestion. I will present why. Microbial electrolysis cell is a second technology. Dark fermentation can be coupled with microelectrolysis cells also. Photofermentation, that can be coupled with dark fermentation, and also others, mostly some algae or dark fermentation coupled with other algae technologies. And so the articles mostly are dedicated to dark fermentation and dark fermentation coupled to anaerobic digestion, MEC in blue here, and little bit of photofermentation, photobioprocesses. And now, if we look at the patterns, the dynamics are a bit different because we have dark fermentation, the coupling, but more MEC and dark fermentation and MEC coupling. And what I'm going to present is this kind of coupling is a key aspect to valorize the waste and the organic matter. And it's also what we are addressing in the BIOCTANE project, how to improve the carbon efficiency of this system. So first, the dark fermentation, which represents about 75% of the publication. What it consists in is we put the organic waste and the effluent into closed systems, and the microbial communities here are degrading the organic matter into hydrogen, carbon dioxide, and organic acids. Some alcohols, of course, microbial biomass is produced, so the microbes are reproducing, and some organic matter can be undegraded because it's not biodegradable. So if we look at the anaerobic digestion process, which is the final product, the methane here, from the carbohydrates, we have a first step of hydrolysis, microbial hydrolysis. Then with the sugars, complex sugars, we have a fermentation step here, acidogenesis. And these sugars are converted to acetate metabolites, so some carboxylic acids, ethanol, and hydrogen. Among the metabolites, what we call metabolites, VFA, we have propionic acid, and we are interested in this specific molecule in BIOCTANE project. And then all these compounds, by acetogenesis, are converted to acetate or hydrogen, and then methanogens can convert acetate and hydrogen to methane. So we try to stop this reaction and only focus on hydrogen and molecules production. So in natural ecosystem and anaerobic digesters, so in nature, hydrogen is produced and the molecules, but they are immediately consumed by methanogens or other microbes. So one of the challenge is really to stop the side reaction and these reactions that are consuming the product. So in dark fermentation, we have few operating conditions that can be controlled: the hydrogen retention time, the pH, the temperature, and the microbial community. So just as a matter, an example, if we take the same degradation scheme, here, the main microbes that are producing hydrogen and other molecules, this one, acetate and butyrate, are Clostridium species, and they have metabolic limitations. On top of that, with waste, we have ecological limitations, meaning that one of the challenge is to deal with different types of microorganisms that can produce a lot of different molecules. And if you look at propionate, for instance, there are few bacteria that enter into competition to degrade carbohydrates. So one of our challenge really in BIOCTANE project is to convert these carbohydrates towards propionate mostly, and try to avoid the side reaction. Yeah, I will just keep this. And in dark fermentation, what is interesting is finally, if in the literature we look at the different substrate, and some substrate like simple sugars, industrial effluent, agricultural residues can have interesting hydrogen yield and also molecule yield. And one of the key aspects is we check the operating condition in all papers. And finally, what we found is hydrogen yield, but it's also the biomolecule yield, were quite similar finally. So if you look at here, the distribution, so of course we have some outliers here, but the distribution, whatever the condition, mesophilic, thermophilic, the volume of the reactor, or if it's operated in batch, fed batch, continuous, semi-continuous condition, we found almost an average value of conversion into this product. That means that under certain conditions, whatever the temperature, working volume, or mode of operation, is quite a robust process. So we just have to find the good microbial communities and the good condition. And finally, the technology will be robust to convert the waste to what we expect in terms of molecules. So the main advantage of dark fermentation is a high organic load, is a well-proved technology, so it's already used to produce to treat waste and wastewater. But the main limits are the metabolic limitations. So the maximum theoretical yield for hydrogen is 33%, and the average yield in literature found is about 11%. And the remaining are the molecules. So that's why it's interesting in a BIOCTANE project, is because we will use the hydrogen, but also the other molecule like propionic acid. Indeed, if we look at in the literature the hydrogen yield and the concentration in organic matter, when we increase the concentration, the hydrogen yield decreases. So there are a whole unvalorized organic matter here in form of hydrogen, and it's mostly recovered in form of biomolecules. So what are the biomolecules? In literature, we found that, of course, acetate and butyric, because they are linked to the hydrogen production, ethanol, and propionic acid. So it is one of the top molecules, often found in mixture with the others. So in BIOCTANE, the most difficult will be to have this molecule very specifically produced compared to the others. So also lactate, valerate, and so on, a lot of different molecules, but often found in mixture. And we did also a market study on these different molecules, and the propionic acid, again, is quite interesting molecules for the green chemistry. So it's also a secondary outcomes of the project that can be used as a chemical for the green chemistry. So indeed, as most of the organic matter is found in molecules, so we can extract, but we can also valorize the molecule. And there are two types of valorization. One is to couple dark fermentation here. We recover hydrogen, and the molecules are converted to methane in the second step in anaerobic digestion. But we can also couple dark fermentation with what we call bioelectrochemical system or microbial electrolysis cells to produce hydrogen and also to degrade or to convert the molecules to hydrogen. Is what I'm going to present. So first, the coupling between dark fermentation and anaerobic digestion is 10% of the articles produced. And I just would like to present some results we got in a national program. We coordinated about biohydrogen production from waste. So toward the implementation of additional fermentation step into AD treatment stream. So the idea here was to study the impact of this fermentation process into the existing AD process to produce hydrogen, but also molecules here. And as a rough example at pilot scale, what we found is the condition where here food waste, so I'm sorry it's in French, but food waste and manure, and fermented food waste and manure. so after fermentation, and here only along anaerobic digestion process. So here are the organic loading rate. So of course, in the two-step process, we had hydrogen first and then methane. In a one-step process, only methane. And if we combine the two-step process, the energetic yield increased by about 40%. It's explained mostly by here. If we look at the error, the methane content per gram of substrate is very similar. So the two-step process, we are used to say that hydrogen is a bonus when we operate a two-step process, because in the first stage, we degrade the organic matter. So it's very facilitated here to produce methane. So the methane yield was almost similar in all cases, an increase of 40%, about 40% the energetic yield. So that's really a key aspect of the coupling. And also in dark fermentation, if we add this step into AD, we look at the LCA. Again, I'm sorry, the description is in French here, but the LCA is very favorable. So the scenarios of the project here, we produce hydrogen at below 2 kilograms of CO2 per kilogram of hydrogen. At European level, hydrogen below about 3 kilograms of CO2 per kilogram of hydrogen is considered as a green hydrogen. So here, with a mix of electricity in France or photovoltaic solar panel in France is below 3 kilograms. Here is a reforming of the natural gas is about 10 kilos of CO2 per kilogram of hydrogen, and the mixture, if we consider the mix, electrical mix in Europe, it can goes up to 20 kilograms CO2 per kilogram of hydrogen, even if it's by electrolysis. So, so it's just to show that the green hydrogen can be produced with this kind of technology. And the most impacting actually is not the technology itself, is the epuration mode. Here was a PSA system. And the impact also on heating on site. So here we use the biomethane. The second technology is the microbial electrolysis cells. So it consists here on the conversion of organic acids, so propionic acids, butyric acid or acetic acids, alcohols, into electrons and carbon dioxide. So we use the capacity of microbes, electroactive microbes. They degrade the organic matter here and they give electrons to a circuit. So it's really in between electrochemistry and biology, microbiology. And so these microbes are providing the electrons, and by applying here a difference of potential, the current is generated and the protons are converted to pure hydrogen here in the cathodic compartment. So what is interesting here is two things. Is we can treat and valorize the organic acids coming from the dark fermentation, and also we produce a pure hydrogen here in the second compartment. So for that, we use different types of microorganisms, electroactive microorganisms that can transfer their electrons to the anodic electrode here, the anode, through mediators in the bulk, or by direct contact with the anode. So through here a cytochrome or with a PD. So what is interesting, there is different types of electroactive bacteria, and is something we will focus on in the BIOCTANE project. And so why do we use bioelectrochemical system and not electrochemistry? If we look at here the cell voltage, theoretically, to go from water in electrolysis to hydrogen, the difference of potential is between 0.2 and minus 0.4 for hydrogen. But if we, at the end, we come from acetate, the difference of potential, it's only between minus 0.28 and minus 0.4. So minus 0.3 and minus 0.4 for hydrogen. That means that in this microbial electrolysis cell, most of the energy comes from this short carboxylic acid. And the difference of potential, it's only theoretically of 0.135, and in the electrolyzer is 1.2. So it's 10 times less than in the electrolyzer, actually 8 times less. And we often put a potential of minus 0.2 to the anode. So this technology is quite interesting because we have a high hydrogen production yield, high energetical conversion yield, and the hydrogen here is highly purified in theory. So it's adapted to wastewater or effluent coming from the fermentation. It has a high material cost and a low organic load. So in previous study, we did this coupling. So you have here a reference. We did some coupling. So we used 6 different wastewater and industrial byproducts, well wastewater, and we tested the feasibility of hydrogen production in a 2-step lab scale process. So in the first step was the traditional dark fermentation process, and in the second step we use the microbial electrolysis cell. So I'm going through this 2-step process. So the dark fermentation, here you can see some yields. So it's very variable according to the type of substrate. So one was cheese whey, wastewater from industrial fruit juice, this one, fruit processing wastewater, paper mill wastewater, sugar production wastewater, and business residues also. What is interesting is in the second step, so we use the effluent here. In the second step, we recovered a lot of hydrogen. And if we consider the energy we put into the system, we recovered up to 700% of the energy put into the system into hydrogen. And the treatment, because it's we have to keep in mind that is a treatment process. The treatment was efficient between 60 to 70% here, COD removal. So as a summary, in this study, we found that in terms of yield, the theory is about 12 moles of hydrogen per mole of glucose, and we reach around 10, bit less, mole of hydrogen per mole of glucose equivalent with this wastewater. So it's about also 70% of COD removal. So it was quite efficient. We were quite surprised to produce hydrogen. So it's a high hydrogen production in MEC than in dark fermentation alone. So it's about 6 to 15 times more hydrogen in MEC than in dark fermentation in the coupling system. So how we can apply this knowledge to the BIOCTANE project? So how we plan to do what we are doing is here, doing a dark fermentation process. So here we will simplify the system, the organic waste, into metabolic products. So as I showed, is a mixture of acetate, butyrate, a bit of hydrogen, and propionic acid. And what is interesting is in the MEC, the electroactive bacteria are not degrading the propionic acid. It's quite difficult to degrade this acid. So they are degrading, and we will shape the microbial community to degrade only acetate and butyric acid. So we will valorize most of the organic matter going through this 2-step process into propionic acid. So the effluent will be enriched in propionic acid and into hydrogen. That can be used in the second step. So the experiments are ongoing. So in conclusion of my talk today is environmental biorefinery. So is anaerobic digestion version 2. We think that will be dedicated to new high-value molecules, especially biohydrogen and bio-based molecules that will be produced from different categories of waste. In BIOCTANE, we focus on mostly bio-waste and food waste. And we couple dark fermentation and, not AD, sorry, microbial electrolysis. And we think it's a promising way to produce biohydrogen at high yield, but also to enrich the effluent in propionic acid. So it's what we call polishing system to polish the effluent and increase the selectivity of propionic acid. And there is still a large field of investigation in this system, in bioelectrochemistry, coupled with fermentation. And because it has a great potential to overcome some thermodynamic limitations that the microorganisms cannot do it. So I hope my presentation was clear enough. If you have any question, I would be pleased to answer your question. Thank you.
Thank you very much, Eric. That was really insightful. And as for the other webinars, again, at least for myself, a new look into some, for me, very wide maps. So a lot of new insights for myself here. As mentioned, if you have any questions, you can either write them in the chat or in the Q&A function, or just raise your hand and elaborate a bit more in detail. In the meantime, I would mention one more organizational aspect, because we can provide you a certificate of participation if you want to receive such a certificate. Just send us an email to info@bioctane.eu. I will also post the email address in the chat in just a minute. And as I said, there were a lot of new things for me here, Eric, so I have some questions as well. You mentioned that in some fields or for some experiments you applied, I think, dark fermentation or even the combination with the microbial electrolysis cell already. So at which scale are those applications currently, and to which scale do you imagine they could develop? You're on mute.
My micro.
Thank you.
Thank you, Gunnar, for this question. Our experiments were lab scale experiments. There is a big challenge in upscaling these processes, because most of our colleagues who did it were facing methanogenesis at high scale. So I would say some groups are upscaling this technology. So I know some of them in Barcelona, especially. But it's quite difficult because rapidly you can have methanogens in this system. There are some examples of pilot plants or demo system. Meanwhile, is still, we have to keep in mind is a treatment process first. So to answer your question is really a hot topic now, is how to upscale this kind of process, definitely.
Okay, so it's still an open question, like how far will we be able to scale it and when? Yeah, very interesting.
When is difficult to answer, but how far? Some groups are investigating this aspect. When is more difficult, because if we want to produce hydrogen, economically now, bioprocesses are competing with electrolyzers. And you know, it can be a bit difficult to outcompete the electrolyzers. So just as a matter of example, is the target is €5 per kilogram of hydrogen after production, the cost of the production. For natural gas reforming, they can reach about €1.5 per kilogram. And for electrolyzers, it, depending on the size, can be a bit below 5 to 10.
See, so
or higher.
yeah.
And if we focus on biological processes, they are not very productive. So for fermentation can reach about 5, but for the quantity is not a lot.
Mm-hmm.
And for bioelectrochemical system, it can be around the same as electrolyzers. So the benefit is really, or higher price, the benefit is really the treatment system.
Yeah, I see. Yeah, that was another question I had, like a cost comparison for different hydrogen provision pathways. As we probably all know, that hydrogen has been quite a hot topic in the recent years. So if I get you right, it could be in the same cost range of electrolysis, for example, most likely not in the range of fossil-based processes. And still, I assume that a lot of development will take on for biogenic hydrogen. So there's probably a large variety also on the cost estimates so far.
Yeah, exactly. The cost estimates are based often on pilot scale, or so you know that we are making a lot of. It's difficult to estimate at full scale what
Mm-hmm.
would happen,
Mm-hmm.
and also because the technology is not implemented at full scale.
Yeah.
So in our estimate, depending on the scenarios, is very sensitive to scenarios, type of waste, hydrogen yield, the conversion yield, the type of pretreatment, and so on. So it's very sensitive to the scenario,
Mm-hmm.
where it will be implanted. It can be competitive, a bit more than €5 per kilogram, but can reach also very high price.
See.
Up to, so I've seen in some literature, up to €50 per kilogram.
Mm-hmm.
So I'm very careful with this assumption, as you imagine, but it's a challenge for the future. But we have to keep in mind is the treatment process, really. So it's the services to the society first.
Mm-hmm.
And if we can benefit from these services, or if we can lower the cost of the treatment, is also part of our development.
Yeah. You mean, in a sense, it's a two-folded advantage. On the one hand, we have a waste treatment, and on the other hand, we get valuable products from that treatment.
Exactly.
Yeah.
The main disadvantage of biological processes is the productivity, the efficiency of the system.
Mm-hmm.
Of course, microbes or nature, we are quite the microbes are quite slow compared to chemical reactions.
I see. Yeah. Beyond wastewater treatment, are there other applications where you could imagine that biohydrogen production would make sense?
Good.
I mean, we're kind of losing one of the two advantages then, right?
Yes, exactly. Good question. I wouldn't, as I presented, only a tiny part of the organic matter is converted to hydrogen, 30% maximum. The others are molecules. So if you look at highly degradable substrate, it will be mostly converted to molecules. So we have to first to focus on these molecules. And this is what we are doing in BIOCTANE. That's very interesting because we are focusing on molecules that will be further converted to jet fuels. So it's highly valuable. We treat and we valorize. if we focus only on biohydrogen, only 30% of the capacity of the potential of the waste or effluent or carbohydrates or any type of organic matter will be valorized.
Mhm.
So for me, it has no sense to say, except if we couple with MEC or other system, but only fermentation, it has no sense to say we focus only on biohydrogen. Let's couple and increase or optimize the potential of the organic matter.
Mm-hmm.
Yeah.
You mean since there's such a broad spectrum of products, and I remember 1 or 2 slides where it's been shown, it makes the most sense where you can use at least several, ideally most of those products and not only the hydrogen.
Yes.
Yeah, and I assume then also the valorization of the entire process turns into something else, because some of those products might be, let's say, more rare or more specific than hydrogen.
Yes, the most difficult part here is purification.
Mm-hmm.
Because when you have a gas like hydrogen, it's quite simple to purify the gas. Methane, hydrogen is not technically so difficult. When it's in the liquid phase, you have a mixture of VFA, of carboxylic acid at low concentration. That's the key point. How can we extract and purify these molecules? Because in the green chemistry area domain, they want pure molecules as precursors.
Mm-hmm.
So that's a key point. That's why also the polishing effect of the MEC would be interesting to be investigated. Because even if it's a low concentration, the selectivity is high, it's always easier to extract or purify this molecule.
Yeah. Okay, that supports more or less in extracting the molecules that you want.
Yes.
Yeah.
You can have.
All right, thank you. I have one more very specific question that you might have answered on one of the slides. So at some point you mentioned at least the way I got it, the energy efficiency of dark fermentation. It showed 3+, which meant advantages to me. Can you compare that, for example, to electrolysis?
What do you mean? I'm sorry.
Yeah. So if the energy efficiency of dark fermentation is rather high, I was wondering, like, where does it range in compared to, for example, hydrogen production from electrolysis or any other, let's say, chemical-based and not biogenic process?
Is actually for hydrogen, it's not comparable because for electrolysis or other system, you provide energy. You have to provide energy to extract the hydrogen. For biological processes, the amount of energy is just mixing and pumping, because the microbes are able to extract themselves the energy. Even mixing is not necessary. So this is not fully comparable because the amount of energy you have to put into the system is very low compared to the other.
Yeah.
But what we saw is when you couple in the anaerobic digestion process, you just implement this hydrogen step, and the two-step process is more efficient than the one-step process. And so the hydrogen is often what we get at lab scale or pilot scale. The hydrogen is a bonus compared to AD. Is on top of that.
Yeah. Okay. Interesting. Thank you very much. Yeah, and what I assume is also another, let's say, second aspect is that in electrolysis you mainly get one product, hydrogen, and with dark fermentation you get a range of products. So how do you allocate the energy that you spend towards those products? If that makes sense.
Yes. So in dark ferment, when we couple the first system, the dark fermentation, doesn't require a lot of energy. Is not the main energy consumption. What I presented, if we only focus on hydrogen, the most energy consuming equipment is purification from CO2. And what is beneficial is we haven't compared, but it would be very interesting to compare in MEC or the coupling, as we have pure hydrogen, to compare the energy consumed to purify hydrogen in dark fermentation that is already purified in MEC. Mm-hmm. So we put a bit of energy, but maybe it's much more less than the purification system. We haven't compared so far, but I think it's a good question. Yeah.
Okay. So yeah, so probably the main or the large energy consumers are other process steps. So will most likely also be interesting to have a closer look then at the results of our work package 4, who are doing the overall process evaluation, not only in terms of technological parameters, but since we also mentioned cost, I could imagine there will be also some interesting figures at some point throughout this project. Mm-hmm. Thank you very much, Eric. Unless there are any further questions, I think we more or less reached the end of our webinar. As mentioned, if you send a short email to info@bioctane.eu, now also written in the chat, we can send you a certificate of participation straight into your mailbox. And apart from that, thank you very much for sharing this insightful hour with us. To let you know, the next BIOCTANE webinar will take place on 6th of June, so another Thursday at 4 p.m. in the afternoon. And the topic then will be focusing not so much on the biogenics, but rather on the thermocatalytical routes, so the use of catalysts to convert the platform molecules that we mentioned, so for example acetoine and 2,3-butanediol, towards jet fuel. So now we had several webinars looking at the provision of those platform molecules, and then finally we'll also turn them into jet fuels on June 6th. You'll receive a registration link and everything else either via our social media channels, or if you have subscribed to our newsletter, you'll also receive an email there. Having said that, thank you very much. Thank you, Eric, for this insightful presentation, and have a nice afternoon.
Thank you very much. Thank you all.
Bye.
Bye.